In this video, we'll be understanding the biotechnology in the field of medicine. Now, our DNA technology or the recombinant DNA technology has come as a great help in the field of medicine. There are many advantages of using biotechnology in the field of medicine. The first and foremost, it helps in the production of the therapeutic drugs in large scale. That means it is capable of large scale production. Large scale production is possible due to this. Second important factor, which is why we are using biotechnology, is the products are safe to use. We can use them safely. The third important factor is they do not generate unwanted immunological responses. They do not generate unwanted immunological responses. Also, no allergic reactions, no allergies. So these are some of the important reasons why the biotechnology is used in the field of medicines. And in medicine, we are utilizing it for the production of therapeutic drugs, in gene therapy, in diagnosis, that is molecular diagnosis. We are using it in various fields. Now, the most important ones I have mentioned over here. Therapeutic drugs are actually very important because these are the drugs which are actually the proteins which are produced from the recombinant DNA technology. So therapeutic drugs are one of the important products which are obtained by biotechnology. And here in this we are going to study insulin, which is a very important drug which is being produced as using the recombinant DNA technology. Apart from insulin, there are several other therapeutic drugs such as that is the growth hormones are there, then the monoclonal antibodies are there, many vaccines are being produced, and several other products are obtained through this. Now, nearly about 30 recombinant drugs are presently being produced, and out of which 12 are produced or 12 are being marketed in India. So we are using this biotechnology in various fields of medicine. Coming to insulin, let us try and understand what is actually insulin. Insulin is a human hormone, and this human hormone is produced in beta cells of the islets of Langerhans in pancreas. It's a human hormone and it's a very important hormone. Now, the major role of the insulin is actually in the metabolism of sugar. So it plays an important role in metabolism of sugar. When we are saying metabolism of sugar, that means it regulates the amount of blood sugar level. Now, if there is less secretion or if there is absence of secretion of this hormone, it results into a disease called as diabetes mellitus. So diabetes mellitus is a disease that is caused due to insufficient production insufficient production of insulin. Now, when the insulin produced is not sufficient enough, or when it is not produced altogether, a person suffers from the disease diabetes mellitus, and diabetes mellitus is very common disease. As a result, the person suffers from many other problems. Now, what was the earlier method of providing the treatment to this disease? Earlier, the insulin was extracted from the slaughtered pig. So the treatment earlier was slaughtered pigs and cattle, and the pancreas were taken from there. Insulin was extracted from there and it was given to the patient. This was the normal treatment given in the earlier times. Now, it had certain disadvantages. The insulin which was produced, or which was used to for the patients over here, differed slightly in the sequence of amino acids from the human insulin. So there was a difference, and as a result of this difference, the administration of this insulin generated certain allergic reactions and immunological responses, so which were not desired. As a person, when it was given, the person was given this, the person suffered other reactions. So the scientists and the team was working for it to obtain the insulin which was similar to that of the human insulin, so that these disadvantages can be avoided. For this, let us try and first understand what is the structure of the human insulin and how it is being produced for the benefit of the human beings. Let us now study the structure of insulin. Insulin actually it is made up of 51 amino acids. The insulin is made up of 51 amino acids, and it is made up of 2 polypeptide chains. That is chain A and chain B. Out of this, chain A has 21 amino acids and chain B has 30 amino acids. Both the chains are linked by disulfide bonds. Now this, we are talking of the structure of insulin. Now, in the case of mammals, that is the human beings we are talking over here, both the chains are produced in the form of a prohormone. So in the case of human beings, when we are talking, it is having the chain, both the chains are being produced. This is chain B and this is chain A. We write it with black only. And this is C peptide. This dash is C peptide. And this is proinsulin. So the body produces the hormone in the form of proinsulin, and it is an inactive form or non-functional. It is non-functional over here right now. Now, the prohormone needs to be processed before it becomes mature and becomes functional. Now, over here, the prohormone, as you can see, is having the proinsulin or the prohormone has an extra stretch of the amino acids, which is amino acid chain, which is known as C-peptide. This C-peptide is actually made up of 33 amino acids. It is made up of 33 amino acid, and this acts as a barrier. Over here, there are disulfide bonds. Now, for the proinsulin to become active and functional, that is, we call the process as maturation, it has to remove this C-peptide chain. So, if the maturation or the proinsulin becomes active, then the process is maturation, and during the maturation process, the C-peptide removal has to take place. As a result, now we have a chain which is having the, over here it's A chain. Now here this will be the B chain, and these are linked with disulfide bonds over here, and we have the C-peptide chain over here, which is being removed. C-peptide. Now this insulin becomes now functional. This insulin becomes functional. Now, so simple terms if we understand, the proinsulin changes into insulin after the removal of the C-peptide chain, which is around 33 amino acids. Now, the main challenge in the synthesis of insulin was to obtain two chains, that is chain A and chain B. We do not require C. So, how it was done? The problem was overcome by an American company, that is Eli Lilly. They came up with a solution in which the two DNA sequences which corresponded to A and B chains of human insulin. So we were using two chains, that is the DNA sequence which code for chain A and a DNA sequence which coded for chain B, were prepared and introduced into the plasmids of E. coli. So these are the DNA sequences which are coding for A and B, and these were introduced in separate plasmids. In E. coli. Now, the A and B chains were therefore produced separately by the two different plasmids. Because they were introduced in two different plasmids separately, they produced A and B separately. Later on, these A and B were combined using disulfide bond. So later on, the disulfide bond was used to create the hormone which was similar to that of the human insulin. Now this human insulin, which was produced artificially using the recombinant DNA technology, is known as Humulin. Humulin is the first therapeutic drug produced by this method, and it is effective for type 1 diabetes and is similar in structure to that of the human insulin. As a result, it is being extensively used because it does not generate allergic or immunological responses. Later on, Brainback C and Eli Lilly produced another better version of human insulin, which is known as Humalog. So now we are also using Humalog, which is having actually better absorption rate. Now, when also one important thing was, now in the insulin, when we are using it, the insulin is cannot be used as a oral pill. We cannot administer it or take it orally. It has the insulin has to be administered in the form of liquid only. That means the reason important reason why it cannot be taken orally is because when it is taken orally as a pill, it goes into the gut. That means that it enters into the digestive system, where it is with a large amount of insulin which is being administered is digested by the body, and very less amount enters into the bloodstream, and therefore it will not be effective for the treatment of diabetes mellitus. So we cannot use insulin as a oral pill. It has to be administered in the form of in the form of liquid only. And here this Humulin, which is being produced using the biotechnology, is similar to that of the human insulin and has given excellent results.